So I'd like to introduce our next speaker, who's Dr. Gaurav Sahay, who's from the Faculty of Pharmacy at Oregon State University in Corvallis, Oregon. And he's been devoting some of his research attention to some of the things that Peter just talked about at the end, and that's the way of specifically associating particular cells and particular tissues. I know I heard him speak about this last year in Vancouver. He gave a great talk, so
thank you. Thank you so much.
Go for it.
I hope I sound all right. Let's see if this works. All right. So thanks so much for the organizers, Beat and Terry, for this very kind invitation. And it's very hard to go after Peter Cullis. So I get the chance of doing that, and I hope I can do some justice. So in our sort of field, before COVID, the idea of using a messenger RNA was that it can very quickly form a protein. It has high efficiency. There's no risk of genetic integration. It does not need to enter the nucleus, and it can be transient and regulatable. And at that point, the hope was that, rather than moving from therapeutics, the field moved to vaccines. And a lot of companies were criticized at that time that why vaccines? They don't make a lot of money. But then a pandemic came and it changed everything. But there's a big challenge for RNA-based technologies, and as Peter said, that these are negatively charged molecules, and you need some kind of an enabling technology that can deliver. And just in 2020, the world really changed. What we used to work on started to come on the New York Times. How these problems like shipping these medicines would be taken care of. And finally, we got injected of some of these lipid nanoparticles ourselves. And I remember having some sort of a fever after getting the injection and dreaming of endocytosis. And I sometimes feel I shouldn't think that much on lipid nanoparticles, but I think they're a fascinating tool. And very recently, we now have a Nobel Prize that was given to Katie Carico and Drew Weissman for modifications of RNA. But I sometimes complain that RNA itself is a dead molecule until it's delivered. And for that delivery, you need this lipid-based nanoparticle that is composed of ionizable lipids that can deliver RNA inside the cells. The Nobel Committee in their advanced sort of report did mention the use of these lipid-based technologies for delivery of RNA. And a lot of fantastic work has been done by Peter and others also in the field now to understand the structure of these ionizable lipids, and initially from knowing little to now know what kind of linkers we should use, what kind of hit groups, what kind of tails can enable delivery has been really fascinating to see as well. And now I'll not go too much into how lipid nanoparticles are formed. This has already been discussed. But I'll say some key features are, especially against like using an AAV, you have a packaging capacity inadequacy. With lipid nanoparticles, you can encapsulate as big as 10 kb. Modularity, you can deliver sRNA, mRNA, now the CRISPR technology. Scalability means a completely different thing now because you could scale for billions of people. All you need to do is find the right lipid. If we can find the right lipid that, like Peter was saying, can go into the heart, the eye, the lung, scalability, and then taking it further is not an issue, which is very unusual for a field. So the rate-limiting step is not that anymore. We know how to make these. Companies know how to develop these as a clinical product. I also would like to say that sRNA, mRNA, and CRISPR all have one thing in common. All won the Nobel Prize. That they have the second thing in common, that they are delivered by LNPs. And I hope one day my friend in the previous talk deserves a certain recognition. The one thing that, again, some folks outside the field, not the nano field, think of these technologies are that these lipid nanoparticles are just for encapsulating. And I challenge that thought because and we can encapsulate out of the libraries that we make, hundreds of lipids would be 98% efficaciously encapsulated. The challenge is to have the right lipid that could, once injected, go into the target tissue, release the cargo in an endocytic vesicle much rapidly, and then disengage and do its action. And that whole action is very difficult, and there are only few materials that do it. And in my view, these ionizable lipids are endosomal escape agents. Many people say, hey, why don't you put like a virus or something like that? Actually, they make these things worse because they themselves are a very potent endosomal escape agent. And this is just one example of a galectin 8 cell line, stably transfected GFP. You throw in an LNP, you'll see those punctate structures. That's basically endosomal sort of disruption and a way to visualize endosomal escape because it's a rare event. You can't usually visualize it. This is one more example where we used a lipid nanoparticle with a cytosterol, and you see this massive escape. So this engagement with an endosomal body makes it an active participant and not just an excipient, like many people think. So where are we going next after this much sort of like success in the field? We can hit the liver, we can hit the muscle, but I think where we need to go next is to deliver into other types of organs from the brain, eye, lung. And one area which my lab is working on is delivery of lipid nanoparticles to the eye. The eye is a beautiful organ. It has this retina which senses light. Light passes through from this NFL towards the photoreceptors, which change the light into an electric signal, and then the brain processes it. If there are mutations in that top sort of panel in the retinal pigmented epithelial cells or the photoreceptors, you start to see disintegration. So this is normal retina. The green is photoreceptors. If you have mutations there, you start to lose those photoreceptors. And most of the mutations are in the photoreceptors. And this is a dog, Polo, from my grad student, Marco, and he just made sort of like a functional assessment. If you start to lose the photoreceptors, you start to get these tunnel visions. And so there are 300 different types of IRDs. The best way to deliver some sort of gene editing tools, like nucleases, would not be through a virus, which will always have an active nuclease, but through a RNA-based system. There is already one product out there through Luxturna. It's an AAV towards the RPE, but again, it's also showing some immune response. You can't control the dose. So we think the lipid nanoparticles might be able to deliver larger genes. So in this case, how you deliver into the back of the eye is two ways. One is the subretinal route. You inject into the back of the eye. You form this bleb. This is done very regularly in the clinic. Basically, clinicians do, like there are specialized clinicians who do this for different inherited retinal degenerations, but it is highly invasive and it detaches the retina. The second way is to give an intravitreal injection, which is more common done in like offices of these clinicians as well. You don't need specialized like clinicians to do this, and you can do like multiple dosing. A lot of diseases like for AMD are being used for that. But the vitreous is viscous. It doesn't allow a lot of nanoparticles to traffic and enter. So what we started to work with was optimizing the lipid nanoparticles to target the photoreceptors with Dr. Renee Rayals, who is now a faculty. She was a postdoc with us, and Dr. Mark Pinesi, who does a lot of clinical trials. He was in the Editas clinical trials as well. And so we thought if we first, in 2018 or 19, we started to just look at the lipids that are already available, like MC3, KC2, and look at where do they transfect into the retina. And what you find here is this AI9 mice. So if you give a clear RNA, you'll see a TD tomato signal, which is red. All the transfection that you see is in the RPE. So RPE is the liver of the eye. So definitely the particles end up there. They don't go into this ONL section, which is the photoreceptor section. If you give it intravitreally, nothing goes to the back of the eye, and it's stuck in sort of the front. So we wanted to deliver technologies that they do two things. One, we want to hit photoreceptors with this technology, and the second is, can we do an intravitreal injection with this technology? So Our recent work, we have started modifying, first we modified PEG, that's already been published, and then we started to use certain PEG variants that give a negative charge to these particles, and this work is about to appear maybe in a month or so. And when we changed the surface modifications of these particles, we found that the positively charged particles were sticking up and were a little toxic in the RPE, but sort of the negatively charged particles, we started to see a behavior that we basically started to hit the photoreceptors. We call this like an LNPX. You still hit the RPE, but these outer segments of the photoreceptors you see very clearly. Finally, like, you know, the eye folks are very skeptical of data. This is the only time when they really started to agree that we can really hit in a very potent way into the photoreceptors. We've also found that using things like MC3 have a toxic effect, even though you think eye is immunoprivileged, but it's not. There is an immune response that can happen. So we started to make a newer library of lipids with this newer sort of chemistry that has been used in small molecules called the Gewald reaction. And what you can do is you can have this thiophene core, and you can change the direction of where you put the ionizable lipids, versus the ionizable sort of head groups. And by changing that, we find that you can also change the tropism, towards the eye, towards some cell types, change the lipid in a different direction, and you start going towards the lung. And this work would come out soon as well. I apologize for this image. I should have given you a warning. This is an injection into the non-human primate using a lipid nanoparticle, which we had developed. And what we find is that in a non-human primate, once we inject these particles, we start to see at a low dose expression into the photoreceptors. But if you use an immunosuppressant, the expression actually goes in and does better, but even at a low dose, you see a very potent expression. And this is the image which I showed Mark, and then he decided that he should be part of a company that we started, and he's a co-founder in a company that we started called Enterex, where in a non-human primate, you see a very clean expression in the photoreceptors, and it lines the whole photoreceptor layer. So I think this is I don't think we have seen such kind of images, at least in public, using non-viral vectors. So we are very excited about that in an NHP model. As I said, MC3 gives an immune response, but these materials do not. We also want to go into the vitreous. So what we did was we've injected a phage library into the vitreous, which had like a 7-mer library. So they are 1 billion bacteriophages with different peptides on them. We injected them into the vitreous, isolated the retina, identified the peptides, put a dye on them, and found that those peptides now could cross and reach to the back of the eye. And we basically have decorated those with the lipid nanoparticles, and we do find that we can hit the photoreceptors with these new modified LNPs, which already are tropic towards the photoreceptors, but now they become even more engaged. We are using human patient-derived retinal cells or retinal models, especially from children who have this disease that will cause blindness. So we have those patient populations. We grow those cells. The pluripotent stem cells are divided into retinal cells, and we find that some of these particles can hit the photoreceptors. And now we are moving into the sphere of gene editing. We are including gene editors like RNA and with the guided RNA, and we have found that we can basically, right now, with some of our materials, hit the RPE really well. We're still working on the photoreceptor side, but we can get gene editing. So we are incorporating multiple RNAs now. And in a cell line level, we are doing something like a prime editing that can also replace certain genes. And we are moving, and we have a grant to push that to different animal models within the eye as well. Finally, I have 1 minute left. There is also an interest to deliver into the lung, and I know my friend Dan Siegert has some outstanding studies on the lung. But one thing which we wanted to do is, with these lipid nanoparticles, the traditional ones, if you use a nebulizer, they sort of break apart. And so you can do formulation changes, and we have done that and published it. But you could also play with the device. So we have basically, with the traditional particle, you lose the RNA, and the particle basically falls apart if you use a vibrating mesh nebulizer. But what we are using is now a microfluidic platform with a company, again, that we co-founded called Rare, which has the ability of a chip that can using microfluidics, form a bubble, which then forms a plume. And the key here is that we have cartridges with different types of sort of places available. So you can put 10 different drugs plus the LNP together. You could have 5 different LNPs in one sort of cartridge, and that will form a plume. So we are very excited to push that forward. But what we see with that is that if you use this microfluidic device, you can save on the size. You can have less polydispersity pre and post. You can have the zeta potential basically the same as is similar, but the RNA encapsulation also remains much better and retained. And finally, you could use it in an animal model, in this case a mice, and you see some good gene transfection. So we are currently working on with these technologies. And one last slide, I think where we are going as a field is to now combine a lot of different technologies. First, I think we can make LNPs now in like 384 well plates. One other transformational thing that has happened is the use of barcodes. You can put those barcodes together, and some of our studies have used this. That will be coming out. You can then target them and inject all of them, or 30 or 40 of them in one particle, in one animal, and then do some sort of an evolution. So we have proposed in a review that would be coming out that we could do some sort of, not to the level of an AAV, but we can have the beginning of a directed evolution for these nanoparticles, because now targeting has been there for a long time, but there is new technologies that are coming in that can enable us to do it in a much more faster, rapid fashion. We have started a center to do GMP manufacturing in Oregon with these technologies, companies that can enable it further. Thank you to my collaborators. Also, we're working with Robert Luxenhofer, and hopefully we'll have some publications with him out soon as well. Yeah, thank you, and questions later, perhaps.